fatica se lo ha fatto toccare con mano. Padova, la sua università e i suoi scienziati sono in prima linea nella ricerca e con questo accordo di cooperazione internazionale ne sottolinea il valore. Fare rete, sviluppare ricerche congiunte tra i nostri laboratori e quelli canadesi, confrontare studi e opinioni tra ricercatori è sicuramente un'opportunità che darà ulteriore sviluppo delle attività di ricerca che si svolge a Padova e nel Veneto. Le ricerche sul genoma sono, secondo gli studiosi, un ambito estremamente promettente per il futuro per affrontare molte patologie. Per chi, come me, è assolutamente estraneo nell'ambito della ricerca medica e scientifica, è affascinante apprendere di ricerche incentrate sulla rigenerazione degli organi, tema di questa prima giornata, e così come terapie genetiche di trattamento delle malattie rare e di applicazione dell'intelligenza artificiale della genomica. E il bello è che tutto questo non è così lontano nel futuro, ma al contrario è un obiettivo che i ricercatori raggiungeranno a breve. Padova ha sempre sostenuto le iniziative di ricerca e studio nate e cresciute nella nostra città e sarà sempre al fianco di iniziative come questa che valorizzano l'eccellenza dei nostri ricercatori. Dobbiamo far conoscere queste realtà, questi programmi, questi scienziati, far comprendere anche fuori dall'ambito medico-scientifico l'importanza e la qualità del loro lavoro e le ricadute positive che potrà avere sulla salute di milioni di persone. In questi ultimi anni abbiamo assistito, purtroppo, al crescere di un irrazionale e pericoloso sentimento antiscientifico. È una cosa paradossale. Se pensiamo ai risultati che la scienza e la medicina in particolare hanno ottenuto in questi ultimi anni. Anche aver sviluppato in tempi così rapidi rispetto al consueto, ma senza venire meno alle verifiche necessarie, i vaccini contro il Covid ne è un esempio. Far conoscere la realtà delle ricerche è un ottimo modo per contrastare lo scetticismo e la differenza tra chi probabilmente ha solo paura di ciò che non conosce. Per questo apprezzo che abbiate organizzato questa Winter School, ovviamente pensata per medici e ricercatori, in modo che sia aperta anche a studenti e a tutti coloro che vogliono capirne un po' di più. Buon lavoro quindi, grazie alla venuta a Padova buon lavoro e ringrazio veramente di avermi invitato oggi. Grazie.
È con piacere che porto i saluti da neopresidente del Coris a tutti coloro che sono qui convenuti e a coloro che dal Canada ci stanno seguendo. È un'iniziativa molto importante perché ci permette di porre a fuoco forse 3 elementi importantissimi che ritengo siano fondamentali nell'ambiente della ricerca, nell'ambiente sanitario per il futuro. Il primo è il confronto. Confrontarsi in modo trasparente, in modo continuo, in modo aperto alle idee, anche a poter dire, beh, forse devo rivedere qualche cosa, porterà sicuramente ad avere dei risultati che porranno il presente in una situazione di grande vantaggio. Il secondo è la ricerca. La ricerca perché il ricercatore non guarda al presente, ma guarda al futuro, usando il presente per migliorarlo. E questo è quello che noi auspichiamo con le attività di Coris, di poter in questa occasione, ovviamente con Genome Québec, ma anche in altre situazioni che stiamo sviluppando, poter potenziare questo spirito di confronto e di ricerca. L'ultimo punto, ricordiamoci che stiamo parlando di un confronto e di ricerca in ambito sanitario. Ambito sanitario vuol dire guardare alle persone. È la cosa fondamentale, ricordarci che tutto quello che noi oggi facciamo, quello che noi stiamo elaborando per il futuro, è un futuro che riguarda le persone, non riguarda le cose, ma riguarda ognuno di noi. Quindi il confronto sulla genomica, sulla intelligenza artificiale, sulla ricostruzione e rigenerazione degli organi è una cosa importantissima perché apre frontiere nuove. Ricordiamoci però sempre che il confronto deve essere rispettoso di quello che è il singolo individuo. Quindi l'augurio è proprio questo. Coris si impegnerà a far sì che il confronto nazionale e internazionale sia sempre più ampio, sempre più costruttivo e sempre però all'interno di ambiti di rispetto, e sono certa che tutti gli scienziati e i ricercatori che rappresentano le eccellenze del Veneto in questo momento e nel futuro ne sapranno dare ampio ed eccellente esempio. Buon lavoro per questa nuova esperienza che spero non sia l'ultima.
Thank you, President. Thank you, Mayor. Now, a few words just to introduce this winter school. The winter school will run from today till Friday. At this time, 3 p.m. local time, with 5 different tables, 1 table each day, where top researchers from both institutions will present their projects trying to find mutual interest. They will cover different topics like gene therapies for rare disease, biobanking, artificial intelligence applied to genomics, and of course, COVID-19 pandemic. And now, before to pass the lead to Dr. Assunta Fabozzo to lead today's table, which is going to deal with organ regeneration and inflammaging, it's my pleasure to invite Dr. Daniel Corder, who is the CEO of Genome Quebec, to give his welcome. So welcome to Padova, welcome to the Winter School, welcome to Science.
Grazie, Professor De Rosa. Mr. Mayor of Padova, Dr. Patricia Benini, President of CORIS, Professor Gino De Rosa, Scientific Director of CORIS, thank you very much for this invitation and organizing this important meeting with your researchers. Buongiorno a tutti, sono particolarmente contento di essere presente con voi oggi per inaugurare questo incontro scientifico. Très heureux de vous accueillir aujourd'hui en cette première de l'école d'hiver entre les chercheurs de nos 2 régions. Il est vrai que le nom de Winter School est tout à fait approprié aujourd'hui au Québec. Il fait moins 17 degrés Celsius. Donc, nous aurons des chauds discours aujourd'hui, malgré la température très froide. J'aimerais remercier, si vous le permettez, Fabio Mincato et Maître Riccardo Donato Meneghetti pour l'initiation, je dirais, de cette coopération entre nos 2 régions. Je les remercie infiniment de nous avoir mis en contact avec le CORIS et pour en arriver à une collaboration qui se concrétise aujourd'hui dans ce Winter School. J'aimerais également remercier Dr. Carole Jabet, qui est directrice du Fonds de recherche du Québec en santé, qui, avec son équipe et notre équipe à Génome Québec, présidée par Stéphanie Laure-Fontaine, ont pu organiser ce colloque. Et bien évidemment, merci beaucoup à l'équipe du CORIS que je salue et remercie également. Cette rencontre de haut niveau concrétise, comme le disait le professeur Geros, une entente de partenariat entre le consortium CORIS et Génome Québec. Nous aborderons au cours de la prochaine semaine 5 thèmes qui font la force de la région vénitienne et du Québec dans le domaine de la santé, des thématiques qui sont majeures pour la suite des soins de santé à travers le monde. Génome Québec a pour mission de catalyser le développement et l'utilisation de la génomique au Québec, et il est très important pour nous d'établir les ponts internationaux qui sont nécessaires au développement de cette technologie de rupture. La génomique et son interface avec l'intelligence artificielle sont à la base de la médecine de précision qui permettront une amélioration majeure des soins de santé. Nous sommes donc très heureux de la tenue de cette première rencontre, porteuse de collaboration scientifique et d'amitié entre le Québec et l'Italie. I wish you fruitful discussion this week, and thank you very much for your participation. Thank you.
So welcome all. Thank you for attending this first session of the Winter School 2022, focused on organ regeneration. I'm going to take very few minutes just to introduce the topic that is, as you can imagine, very wide in terms of fields of investigation. And it will range from the analysis of satellite cells, stem cells inside tissue, through the evaluation of novel technologies and platform in vitro, such as 3D platforms, or also microfluidic high throughput platforms as well. And we will end up analyzing the effects that we can predict in vitro models, as for example, organoids, by also administering nano devices such as EVs charged with targeted mRNA. At the end, finally, we will see the relationship, we analyze the relationship between aging, senescence, and inflammations, and we will cover very wide aspect of the organ regeneration topic. I don't want to waste time and take time from the speakers that are here today, physically and also virtually, and so I'm leaving the words to them, asking them kindly to stay in the 10 minutes presentation we have reserved for them, and we can have at the end a break of 10 minutes each, and afterwards we can leave time for a longer discussion to go deeper into the topics. So our first presentation is done by the youngest speaker we have in this session, and is Dr. Anna Maria Tolomero. There is a research fellow in cardiovascular regenerative medicine laboratory of Professor Gerosa. And so Anna Maria, please, you can have your presentation.
Okay. Thank you, Assunta, for the presentation and to give me the opportunity to talk with all of you about my experience. Until now, I work on the regeneration of 3 different organs. I started with gut during my PhD, and specifically, I work in the field of the inflammatory bowel disease, for which until now, no curative therapies exist. But recently, the mesenchymal stromal cells received the approval from the European Agency for the treatment of the inflammatory fistulas in the Crohn's disease. For this reason, together with Professor Muraka, we decided to test the vesicles, because the immunological effect of the cells is mediated by parachnid factors veiculated from the extracellular vesicles. For this reason, we decided to test these vesicles in an animal model of experimental colitis induced through the overexpression through the administration of DSS. And in this case, we demonstrated how the extracellular vesicles were able to induce the reduction of the severity of the colitis in the animals, but especially to induce the regeneration of the tissue. The second organ is the brain, because today a large amount of neurological disease studies have been performed on biopsies taken from cadavers, or when the disease is at the end. So you can understand that in this situation, it's impossible to study the development of the disease or to understand how or when the disease happened. In this way, next. Okay, here. But we have the in vitro models that could help us, because we have the opportunity to work with the stem cells taken directly from the patients. For this reason, together with Professor Vassore, we define new protocol for the neural generation, taking advantage of the integration between transcriptional programming and the microfluidics. For what concerns the transcriptional programming, we We use the mRNA encoding for engine 2, the neurogenin 2, a proneural gene essential for the correct brain development. And with these technologies, we define a new protocol for the neural generation. And in only 10 days, we obtain two different cell populations, the neural progenitor cells and the neuronal cells. And we obtain only 10 days using only one transcriptional factors. And lastly, I tried to put together all this expertise in the regeneration of a third organ, the heart. And in this case, together with Professor Gerosa and Dr. Fabozzo, I'm working on two main projects. The first is related the generation of a personalized biological pacemaker, starting with patient cells. And in this case, we used different technologies like the cell-based strategies and the tissue engineering. We use these cell strategies because we will work with two different cell type, like T cells derived from blood or the skin of fibroblasts. And in this case, we will use also the cell reprogramming technique because in this way we could reprogramming the adult cells in a more embryonic phenotype. And starting with these cells through the cell programming, we will obtain the desired cell type. We use also tissue engineering because clearly we need to create a scaffold for our patch. And in this case, we will use the porcine extracellular matrix decellularized to create like a bioink for the user to using with the 3D bioprinting, because for the node we need to create a more defined, a more precise structure for the patch. And lastly, the last project that I'm working on is the organ perfusion for the organ preservation for the transplantation. And in this case, we use the extracellular vesicles isolated from an in vitro culture of mesenchymal stromal cells as a medical device to protect, to reduce the ischemia damage occurs during the worm ischemia, and especially the mRNA to as a gene therapy to reduce the immunorejection from the host. And yeah, this is the main project that I'm working on. And yeah, thank you.
Thank you, Anna Maria. Yes, your preliminary data are very interesting. We're going to have the chance to comment them later on in the discussion. Definitely, he opens the path to the next speaker. There is Dr. Colin Christ, who is focused instead on a different approach for treating stem cells. While you were talking about endogenous stem cells, iPS reprogrammed, Dr. Christie's focus on recruiting satellite stem cells from the tissue. Please, Dr. Christ, trying to stay in 10 minutes, please. Have your presentation.
Great. Thank you very much for the introduction. So I hope everyone can hear me, and I'm about to share my PowerPoint presentation. Does it look, are we on full screen for my presentation?
See your screen shared.
You can see it fine? Okay, very good. So I begin.
We cannot see it, yeah.
Oh, you cannot see it?
Once again.
Let me see what's going on.
Yeah, now it seems so. Can you hear us, Dr. Christ? No, you cannot.
I can hear you fine.
Okay, perfect.
I don't know if you can see my screen or not.
Yeah, you're moving it. So if you just zoom it out, so we can see just the presentation.
Okay. I'm going to share it like this, then I think that should work. So thank you very much for having me participate today in this winter school, and I'm very pleased to explain to you today our research. So our lab is interested in skeletal muscle regeneration. And on this slide, I'm showing you a transverse section of adult skeletal muscle taken from a mouse model of Duchenne muscular dystrophy. And you can see all of the individual myofibers in bright field. And you can see the regeneration phenotype due to central nucleated myofibers, which are encapsulated by an extracellular matrix, which we can visualize with antibodies against laminin. And the cells that we're particularly interested in here are labeled with antibodies against PAX7 in green. And PAX7 labels all of the adult muscle stem cells that are critical for the regeneration of skeletal muscle. And the regeneration of muscle is very efficient. So here is a cross-section of tibialis and anterior muscle that is uninjured. And in the lab, we use a cardiotoxin injection, which is a snake venom that we inject into the tibialis anterior muscle. And you can see that 3 days after injury, there's very profound degeneration of the myofibers due to this acute injury. But thanks to the activity of the muscle stem cells, by 10 days after cardiotoxin injury, we can see many of the myofibers regenerating, and they are marked by central nucleation. And by 21 days after injury, the myofibers are growing in size, the function and the strength of the muscle has been restored, and this is all thanks to the activity of a rare population of muscle stem cells that activate both the cell cycle and the myogenic program when they receive a cue to regenerate muscle. And so the muscle stem cell is also called the satellite cell for its satellite position around the outside of myofibers. It is normally present in a quiescent state, which means it has exited the cell cycle, and it is marked by transcription factors of the paired homeodomain family PAX7 and PAX3 in a subset of muscle. Quiescent muscle stem cells have also interestingly activated the expression of MIF5 and MyoD, but they only activate high levels of MIF5 and MyoD protein when they activate to regenerate muscle. So initially they enter a cell of or a state of expansion, rapid proliferation to expand the myogenic progenitors that are needed to fuse and differentiate in order to regenerate muscle. And importantly, a subset of activated muscle stem cells self-renew each and every time we injure our muscle in order to restore that population of muscle stem cells for future rounds of injury. A lot of work in our lab has stemmed from our initial observations that in a quiescent muscle stem cell that is marked by PAX7, we can observe many RNA granules in the cytoplasm of these cells. And these RNA granules are full of RNA binding proteins and translationally repressed mRNAs. And we've made several observations observing these RNA granules. And so one was when I was a postdoctoral fellow in Margaret Buckingham's lab at the Institut Pasteur. We showed that these RNA granules are sites of sequestration of MIF5 transcripts that are targeted by microRNA and localized into these RNA granules that are present in the cytoplasm of a quiescent muscle stem cell. When I initiated my own lab here at McGill University, I showed that the phosphorylation of eIF2 alpha, which is a global pathway that inhibits protein synthesis globally, is a translational control mechanism that regulates muscle stem cell quiescence and self-renewal. And eIF2 alpha phosphorylation maintains overall low levels of protein synthesis that is required for the formation of these RNA granules. And more recently, we have shown that the phosphorylation of eIF2 alpha, although it globally represses protein synthesis, it also enables the selective translation of very specific mRNAs. And as an example, an mRNA for TAC3, which is required for proliferation, is specifically or selectively translated when eIF2 alpha is phosphorylated. And so we are building a model whereby eIF2 alpha phosphorylation is required in quiescent and self-renewing muscle stem cells to initiate a global repression of protein synthesis, but also enable the selective translation of specific mRNAs that are required for the stem cell properties of the muscle stem cell. Upon activation of muscle stem cells in order to regenerate muscle, that is marked by the rapid dephosphorylation of eIF2 alpha to restore global levels of protein synthesis. And this process can be inhibited by a small compound called SAL003, which inhibits the phosphatase enzyme that dephosphorylates eIF2 alpha. And with this knowledge in hand, we want to use small molecule inhibitors of eIF2 alpha phosphorylation to generate novel methods in regenerative medicine to treat muscle disease. And in our lab, when we think about these novel methods, we're looking towards growing more stem cells. So can we increase the numbers of muscle stem cells that function as a skeletal muscle stem cell during ex vivo expansion conditions? We're also trying in our lab to create specialized cells. And so we're asking, can we differentiate pluripotent stem cells into a cell that resembles a muscle stem cell? One of the barriers in the field with respect to skeletal muscle is that although we can have very good differentiation programs to differentiate pluripotent stem cells into skeletal muscle, the myogenic progenitors resemble those that are found in a developing embryo rather than adult muscle stem cells. So we want to ask, can pharmacological inhibition of eIF2 alpha dephosphorylation help to mature these cells into a muscle stem cell? And lastly, can we use the phosphorylation of eIF2 alpha as a pharmacological target to promote tissue repair? And so here we're trying to stimulate or restore muscle stem cell function in vivo. And so with the question of can we use the inhibition of eIF2 alpha phosphorylation to grow functional muscle stem cells in culture, I introduce you to the problem when we isolate muscle stem cells from skeletal muscle and put them on plastic dishes and culture them for a very short period of time. The default program appears to be differentiation, and that can be visualized here by these very long myotubes and that are expressing myogenin and troponin T, and there are very few muscle stem cells present in the culture. If, however, we isolate muscle stem cells and culture them in the presence of our small molecule inhibitor of eIF2 alpha dephosphorylation, we maintain high levels of eIF2 alpha phosphorylation and also high levels of the PAX7 positive progenitors, where PAX7 marks the muscle stem cells, and overall low levels of myogenin, which marks the differentiating myoblasts. And to ask if these cells maintain their regenerative potential after a period of ex vivo expansion, we turn to a mouse model of Duchenne muscular dystrophy, an engraftment model where we isolate muscle stem cells from a PAX3 GFP mouse. We culture them in plastic dishes for a period of 4 days to expand them, either in the presence of SAL3 or in control conditions. And then we inject these cultured cells into the mouse model of Duchenne muscular dystrophy. 21 days after the engraftment, we take transverse sections of the engrafted skeletal muscle and look for dystrophin-positive myofibers, which is a marker of stem cell contribution to myofiber size, and also GFP-positive muscle stem cells, which indicate that they have come from the progenitor cell population. And you can see that if we cultured our muscle stem cells in the presence of SAL3 for 4 days, they retain their regenerative properties as visualized by many dystrophin-positive myofibers at the site of engraftment. And this is compared to our control culture conditions where we lose the dystrophin-positive myofiber numbers. And so I'm going to go very briefly because I believe I've run out of time, but we have this system now where we can expand muscle stem cells ex vivo, and we are currently asking, can we expand muscle stem cells in vivo to promote tissue repair? And for this project, we have been working with a chemist at McGill University to optimize the structure for enhanced activity to identify new analog that inhibits eIF2 alpha phosphorylation. We are treating our mouse model of Duchenne muscular dystrophy for a period of 14 days in the presence of this compound. And when we do so, we observe a higher number of PAX7 positive progenitors across multiple muscle groups after an in vivo treatment with our small molecule inhibitors of eIF2 alpha phosphorylation. And I'm going to end with that. These are the lab members that have participated in the project and our collaborators who we rely upon. And these are the funding agencies that support our research. And I've run out of time, so I'm going to end with that. And I thank you for your attention.
Thank you, Dr. Chris. Thank you for staying in time. Next speaker is Professor Annalisa Angelini, Professor of Pathology at University of Padua. Please, Professor Angelini.
Thank you for the introduction, and may I have the first slide? Organ regeneration, a new perspective. So our journey in regenerative medicine, can you go on? Started a few years ago when we had the possibility to study a model of a transplant patient where we use a donor female heart put into a male recipient, and through in situ hybridization, we were able to identify the transdifferentiation of male stem cells into new cardiomyocytes, thanks to the ability of detect the Y chromosome. And this engraftment was a continuum due to the injury that this heart has as a rejection in this model of chronic injury. Next slide. Then we move to an animal model of congestive heart failure, where we injected two different types of stem cells, rat adipose tissue stromal vascular fraction progenitor cells, and human amniotic fluid stem cells. And we evaluate the stem cell engraftment in different organs. You can see here the amount of engrafted cells that you can see in green and red into the heart. Go on, please. But also in different organs. And in this case, we stain the stem cells with von Willebrand factor for endothelial cells and smooth muscle cells for arteries formation, and even in this cell we were able to see the formation of a microvessel, capillary, and small arteries formation. Go on. And this again, you can see with different staining, the possibility to detect the engraftment and the potential regenerative of these stem cells, both for arteries and capillaries. In the heart also, we were able to see only very few stromal-derived GFP cells with a striated pattern and sarcomeric appearance, confirming that few stem cells fully differentiate into cardiomyocytes. And you can see here also with the connexin 43 to pick up the functional junction between two cardiomyocytes. You can go on, but this was really a very rare phenomenon. In the skeletal muscle also, we were able to identify angiogenetic differentiation of this injected cell, and you can see that we were able to reconstitute the number of PAX7 and MyoD positive cells as marker of the niches, the satellite cell in the skeletal muscle. And we were able to increase the number of both quiescent satellite cell, but also activated satellite cells. Go on. In the kidney, besides the angiogenetic possibility of engraftment and differentiation, we are able only to pick up the presence of newly formed tubular cells, mostly in the medulla and less importantly in the cortex. We understood that in our model of heart failure and interaction between the heart and the kidney, cardiorenal syndrome type 2, the stem cell treatment produce improvement both in the heart and in the kidney, not by regenerative of parenchymal cells, but by a paracrine action with an anti-inflammatory and anti-apoptotic effect produced by the small amount of engrafted cells. But let's put things into perspective. We go on. We changed then our paradigm, so we focus on the paracrine action, not just the potential of differentiation of our stem cells. And we understood that the stem cells produce a broad spectrum of cytokine, chemokine, growth factor, extracellular vesicle. And then we focus our more recent research on extracellular vesicle and synthetic nanoparticles to try to modify or to alter the injured cells in the tissue. Go on. So the central of our research is the extracellular vesicles and the nanoparticles that we know can play a major role in tissue regeneration, but also as delivery vehicles or as a diagnostic or cancer immunotherapy modulator. Can go on. So we got the opportunity also in translational medicine to study the model of heart transplanted patients in which we evaluate the potential of extracellular vesicles as a surface marker to differentiate between acute cellular rejection, humoral rejection, and normal patient. And through MFI, with multiplex flow cytometry data analysis, we were able to characterize some markers able to identify the two different types of rejection. And you can see in the heat map where clearly there is a differentiation between the profile of antibody-mediated rejection and the acute cellular rejection. And this was not just one marker, but a pattern of markers expressed on the surface of our extracellular vesicles. Go on. So we applied our results also to a machine learning algorithm to try to find a predictable graft rejection with a double random forest model. And we saw that with this two-step machine learning model, we were able to differentiate between patients that were negative for rejection towards patients with both acute cellular rejection and antibody-mediated rejection, with a good accuracy and a good validation percentage. Go on. So by profiling endovesical surface protein, we are able to discriminate different types of allograft rejection. And so we count on expanding our population to confirm these preliminary data. Go on. Then we can skip this one to the sake of time. And now we move towards the possibility to insert microRNA in lipoic acid nanoparticles, synthetic nanoparticles, which are are able to be internalized, phagocytes, by the parenchymal cells like the cardiomyocytes, besides the endothelial and smooth muscle cells. And go on, the preliminary results were able to show that, for example, on the right-hand corner, you can see the ex vivo macroscopic distribution of the tagged nanoparticles. After only just one injection, you see the red color and the yellow color, showing the presence of the nanoparticle inside the different organs, the heart, the kidney, the lung, the spleen. And in these central images, you see the red small dot spots that are the nanoparticle inside the cardiomyocytes, as well as inside the tubular and vessels. Go on. And these are really preliminary results with confocal analysis and in situ detection, not only of the nanoparticles, but also inside the cell, the yellow dots are the mRNA with conjugated fluorescence. And so the microRNA is able to reach the cells, and we want to modulate the cells through these nanoparticles. And I would like to thank the Padua group that collaborate with us for the production of the nanoparticles and the translational project that we conducted up to now.
Thank you, Professor Angelini. You open us several topics that we can then go deeper into in the discussion later on. So we can jump on the other speaker, special guest, Dr. Professor Thomas Martin Darkin, Assistant Professor at the Department of Neurology and Neurosurgery at the McGill University. Welcome. Can you hear us?
Yes, I can hear you. I'm just gonna.
Please. Okay, you can have your presentation. Please, pray. Just try to stay in the 10 minutes so that we can have a longer discussion later on. Thank you.
Sounds good. Do you see my slides okay?
Sorry?
Do you see my slides okay? And can you hear me okay?
We cannot see your slides, no.
No? Can you see them now or no?
We can see your face.
That's a great face.
Yes.
Let's see, share screen. The Padua talk. Okay,
That's
now?
it. Yeah, we can see it.
Perfect. All right, well, thanks for the invitation to talk today, and congratulations on the 800 years anniversary. So I'm glad I actually could talk about the work we're doing at the Neuro on applying patient-derived induced pluripotent stem cells into better understanding disorders of the brain. And so my name's Tom. I'm an associate professor at the Neurological Institute, and I also direct the Neuro's early drug discovery unit. And what we are as a group, really, with human-induced pluripotent stem cells at our core, we're kind of divided through four main groups. So the first group in terms of phenotyping, generating, and CRISPR editing stem cells. The second group kind of more on discovery assays and screens. And then the third group, which I really focus on today, is how we're kind of making organoids, 3D spheroids, and using kind of tissue engineering to kind of build more physiological models. And this all feeds into our fourth group, which is really focused on training and outreach. So we're a group of about 40 to 45, depending on the time of year, combining both full-time staff and trainees and everything. And we really cover a gambit of activities, really focused on the brain, and then kind of getting into different diseases depending on researchers that come to us. So everything that we focus on really comes from the patients that come to our doors at the Neuro. We're able to consent them, collect blood from them, urine, and then we're really able to then go into their induced pluripotent stem cells. And then from here, we go into developing kind of assays in either 2D cell types, disease modeling, and then ultimately the goal is to bring it then into clinical trials. So we're an open science institute, so we do work about...
I'm sorry, I think we have some problems with your microphone. Is that possible? I mean, is it something of a rumor?
Can you hear it okay? Is there...
Yeah. I don't know if you can stay far from the microphone. Maybe it helps.
Okay, is that better?
Not that much.
Is it like too loud or too quiet?
The point is that we cannot really understand what you're saying. Otherwise,
okay. Let me just check. So you can't hear me properly now, like even with the audio, or?
Yeah.
How does it work if I just talk like this?
Maybe it's better. Let's try.
Can you hear me better now?
A little bit. Try to talk, and then if we cannot really see,
Sure.
you can understand, I'll interrupt you.
How does it sound from here? Any better or no?
Okay, go ahead, Dr. Darkin.
Okay. Just stop us if you can't hear us, okay?
Better than before.
Okie dokie. All right. So the slides, you can see them again okay now?
Yes, we can.
Okay. And I'll try to talk if it's too bad, just, tell us to stop and we can switch it around maybe to a different microphone. So we were talking about the open science. Yeah, so everything we do in terms of iPSCs is how we study brain disease in a dish. So it all starts with the patient going to the blood cell, and then from here towards induced pluripotent stem cell. And our goal is really to kind of characterize 2D brain cells or 3D neuronal organoids. And this is how we kind of make the brain organoids. So we start with the iPSCs. We kind of make this what we call an uniformed embryoid body, so a 3D spheroid, and then embedding in matrigel and ultimately kind of the combination of factors, so chemical, physical factors, then we can go into midbrain organoids. And then these can be grown for anywhere from 1 month up to 18 months. And this is what we use then for kind of studying Parkinson's, developmental disorders. And so why do we study 3D? Well, they're interconnected, so they're like a piece of tissue within the brain. They're scalable, easy to maintain. You have a diversity in cell types. They can be customizable, and then you can kind of use them to test therapeutics, or that is the ultimate goal. And so really, when it comes to organoids, what we've taught is essentially all models are wrong, but some are useful. So this is the quote from George Box. And the reason why we say this is it kind of relates back to the iPhone. And that with the iPhone, it came in 2007. It was the first version, but now 15 years later, we're kind of on version number 29 or 30, where it kind of has an iterative process where you add features onto it. And this is where we're kind of with neuronal organoids, where we're kind of on version 2. And we kind of need to add different pieces to it, because when you look at the brain organoid, it's a kind of small piece of neuronal tissue, but it's missing both vascularization, immune system, and it has a lot of stuff that similar to the brain, but it needs to kind of be more advanced. And how do we get to version 3 and beyond? Well, we've kind of looked at different aspects within our group: bioreactors, fluidics, characterization, better characterization, microfabrication, and other kind of biomaterials. And for today, I'm just going to talk about reactors and characterization. So one area we've kind of worked on is how we can scale to make 15,000 human brain organoids at any one time. So that revolves around the use of what we call embryoid body discs, so with large-scale EB generation, to then actually make organoids at a scale of anywhere from 100 up to 900. This is what like a 3D, a kind of a 3-month culture would look like. where they kind of just shake around and are maintained and grown over time. And from this, we can grow anywhere in our lab up to 15,000 human brain organoids from different cell lines at any time. And so what do we use them for? Well, we're getting them from different patients with Parkinson's, ALS, neurodevelopmental disorders. And we ask, like, what do they look like so we can better understand what the disease profile of the patient is. We look at different imaging modalities, where one is where you can cryosection them, where you can get a 14 micron section. One is where you can just get the morphology. And then the other one is where you can do a 3D optical reconstruction. And so this will involve a kind of process called tissue clearing or cubic, where you can take the lipids and remove them. So you make the organoid opaque. And then from here, you can then reconstruct it. So you can actually optically section through it with a confocal microscope or a kind of automated confocal system. And then you can then reconstruct it here with the different cells and neurons and start to do quantification and place where they are. And the other thing you can do is also ask, what are they made of? So a cellular jigsaw, where you can take them and break them into their individual components or individual cells. And you can also make like a virtual map describing all the different cell types that are there. And this is really work that kind of developed by Relina V and Dr. Joe Stratton in the group, where we're taking different patients into different organoids, combining them into kind of what we call virtual assemblies. And then this is a forming us both when we take the imaging and combining it with the single cell, we can then start to identify different disease phenotypes. And what we're trying to do is really get towards target-driven models, where we start with the patient, work our way through a kind of combination of 2D cell types relevant to the disease, screen small molecules, and then find lead molecules that eventually we can go back into the organoid and ultimately into a clinical therapy. So I'm just going to stop there and we'll take any questions, and I know we have the discussion later on.
Thank you, Dr. Daken. Next speaker is Professor Stefano Piccolo, Professor of Molecular Biology at the University of Padua, who is going to introduce us in the words of inflammaging. Please, Professor Piccolo, have your presentation.
Okay, bene. Thank you. I had prepared something a little bit more specific, but doesn't matter. And the topic we are interested in is in cell mechanics. Mechanics is essentially a general word defining how cells touch each other, how cells perceive each other, not through chemicals, not through hormones or paracrine factors, but rather by physical means, by touching each other. Now, when cells start to touch each other and get, as you can see in the previous presentations, outside of tissue culture plastics, you start to see some wonderful behavior popping up. Now, these are called emergent properties. Now, if you consider cells are part of a community, and all what we discuss here is about regeneration, right? So we have been seeing several aspects of in vitro cultures, still is recapitulating some aspect of regeneration, but it is not regeneration. Now, irrespectively, this is the behavior of a community that cannot be recapitulated by each individual element of the community. So in the next slide, there are some remarkable social behavior, and I think that regeneration is this. You have seen example of organoids, of course, then even contact inhibition or proliferation, and of course embryonic development are great examples. And this is all, they have a landmark, and this is a community effect when forces can flow through many cells diameters. If cells can perceive forces, they are aware of the entirety of the organ in which they live. And so we are interested in this from a very reductionist approach, very molecular approach. And this started from, can I have the second one? Thank you so much. From our discovery that in fact, There is, in fact, nothing really special about mechanical signals being exchanged by cells, but this is just yet another signal. And this is delivered through a cascade through the nucleus, and so that mechanical cues can influence cell fate in a way or another. So we discovered this, and now, thank you, next, this type of sensor that is defined by the activation of two transcription factors is now a generalized view of how cells feel mechanics in many, many different contexts, well beyond those that we had originally described. Next. Now we are interested in YAPTAs as determinants of cell fate plasticity. And next. So this is an example how to make adult stem cells. And we are using the mammary gland as a paradigm. And the mammary gland, as you know, normally regenerate all the time after lactation in normal physiology. And what we discovered, to tell you, to make the long story short, is that next, if you take differentiated cells from any mammary gland, you induce expression of YAPTAs in these cells. And through this transcription factor, you carry out a reprogramming that is very different from the reprogramming you just heard about. It's not Yamanaka type of reprogramming, but it's rather conversion of a differentiated cells into its corresponding stem cells in the living body. Now, these stem cells that are induced stem cells through YAP are next, actually indistinguishable by transcriptomics to the natural basal cells that display some stemness properties, and that are able to, once you inject them into the mammary fat pad of a mouse, to rebuild, thank you, an entire new transgenic mammary gland. So we can regenerate the entire epithelial mammary tree using a single cell reprogrammed through YAP. Next. Now, why is this important? Well, because this is an example of plasticity that relates to tissue regeneration in some epithelia. And this is something that you already heard of, which is induced inflammation in the intestine. Now, in transgenic mice that are conditional mutants, that you can knock out the genes at will, normally you have regeneration, obviously, after an insult, inflammatory insult, but not in the case of a YAPTAs mutant. And the reason is that YAPTAs are absolutely required for making this transition from a normal stem cell, actually from any cell, into a progenitor. The cool discovery here, next, is that although we originally thought that we were converting these residual cells that were remaining after damage into a corresponding stem cells of that tissue, we were surprised to see that in fact this is not the case. In vivo, what happens is that you go into a black box phase in which you have no marker whatsoever of any differentiated lineage of the adult, including the adult stem cell lineages. But for a couple of 2 weeks, while you repair your intestine, and perhaps while you repair any other organ can regenerate, you have a transition to something that does not exist into an adult body. That is, if you want, a holiday moment in which these cells go back to a progenitor state that is a fetal progenitor state. So basically for 2 weeks, after you get an intestinal infection, your stem cells are not those that are normally present in the gut of a healthy individual, but correspond to the same cells that are present during fetal development. And then they kind of age very rapidly after repair is resolved. And what YAPTAs do is, in fact, allowing this transition. So as a matter of fact, we speak a lot about organoids, and it's very known that organoids are not proxy of the real tissues, not only because they miss everything from mesenchymal cells to immune system and go on, but also because, they are not really proxy of the real tissue, but actually proxy of regenerating conditions. And just like, regeneration is key and requires YAP, even organoids require YAP. So in other words, we have settled into next paradigm in which this is work that we initiated this, then many others took successfully the story, and this is work from Prisca Liberali and Matthias Lutolf, who published both in Nature about these stories. There is kind of a transient step toward the fetal stage, that which we find interest in many ways. Next. So you have a backstemness, you have a time machine if you want. In the next. So this is also relevant for transformation. So you have a continuum between regeneration and transformation. That is a unique program that is not shared by healthy tissues. Okay, so healthy tissues couldn't care less about YAPTAZ. You have a nice mutant without YAPTAZ, and they are still functioning perfectly well, organs in the epithelia. However, not if you are challenged the organ to regenerate. That's the bottom line. So the same domain is pertinent to regeneration and transformation. Next. So YAPTAZ are, of course, extremely interesting in the cancer. There is no solid tumor with no upregulation of these factors. Next. And we and others, this is just the selection of what we have done, but this has been done by many others. This is kind of a super hot field. I've shown that there is no cancer without YAPTAZ. We show this in the liver, in the pancreas, in the skin, in the intestine, and the list is much longer. So this is in epithelia, and so many companies are now interested in targeting YAPTAZ as if this next were the magic bullet, okay, for cancer. And we, of course, contributed actively to this view. But we are starting to now to rethink about this paradigm, because we asked ourselves what happens if we carry out the knockout in other contexts. And these other contexts are, next, not only the epithelial layer that we have been studied so far extensively, but next, in the stromal cells. And this connects to this issue of aging. Aging is an enormous problem, obviously. But in the field of cancer, at least this is how we enter that, it's known as the most potent carcinogen. A tumor is massively increased after 60s, the incidence. And so what is happening during this time? Well, that all the connective tissue are severely affected by accumulation of defects in collagens, in stromal cells, and so forth. And this is kind of an Antarctica. Nobody studied anything about stromal cells. So we did an experiment, which is carrying out the knockout next of YAPTAZ in stromals and not in epithelial cells. So what we found, I just follow the white color, that there is a progressive loss of YAPTAZ activity of mechanical properties of tissues as aging proceeds. So we wanted to recapitulate this experimentally, and we carried out the knockout in fibroblasts. And the striking phenotype, next, is that we started to see dramatic multi-organ aging defects, okay? So mice start dying very rapidly after knockout in connective tissues. And the phenotypes are quite remarkable next, because, a young YAPTAZ knockout cells are very similar to old mice And even more striking, if we now boost the expression of YAP, which is a long heroic experiment, you keep on adding some, doxycycline to have YAP expression, you actually prevent aging. And so I think it's interesting, and it tells you something more about the biology of these mechanosensors in living tissues. That's it. Thank you so much.
Thank you. We could hear from you for hours. We can leave some minutes more in the discussion phase. But now I would like to leave the floor to Professor Julie Fredette at the Department of Surgery, University Laval. Good afternoon, or for you, good morning, Dr. Fredette.
Good morning. Let me share my screen. Okay, they say unable to share. I'm sorry.
Actually,
Provided my slides, so I don't know if it would be easy.
Yeah, I think we have a little problem, as we cannot really hear clearly your voice, and also we cannot see your slides. So just to give me a second to understand if it's our problem or not.
Okay.
Dr. Fredette, sorry, we need to redo the connection. So I think we are hanging out and connect once again, just to give us a minute. Sorry.
Okay. No problem. I can try to share, but you also have my slides on your side, so I don't know which one will be easier.
Dr. Fredette, can you hear us?
Yes.
Yeah, okay.
Yes, very well.
Okay. Okay. Do you want to try to share your slides? Otherwise, we are going to share them for you, right?
Please share them for me. It doesn't seem to work on my side for sharing.
So,
Hi, Professor. You have just to increase the dimension of the slides and reduce to icon the browser. This is the browser. You have to reduce
because it's
the icon this browser with the yellow button on the left side.
Okay, because it says cannot share, but you do seem to see something, or?
Well, we are able to see your full screen. So if you reduce the browser,
okay.
Now you have canceled the slides.
And you see slides?
Just try on the bottom part of this of the system. You have the slide screen button that is the third from the left. You have the microphone, the webcam, and the third is the sliding share button.
Yes, I have done it many times and it doesn't allow me.
Maybe you have to allow Google Chrome to screen share your computer.
I'm so sorry. I'm with Firefox. The second one you said working well was Firefox. I'm with Firefox.
Well, it's better to use Google Chrome, as we said in the first email.
Okay. But I provided you my slides. Is there a way for you to share them? And I will just say next.
Just a moment.
I'll use these 2 minutes to ask you, Dr. Ferret, as I saw your slides in advance, if you can leave the vasculature insertion inside the organoids for the discussion later on, so that we can use that as a cue for discussion. Do you mind if we do so? Okay, please, thanks.
No problem. It would have gone faster if I would go through the slide myself, but it doesn't really want to share.
Professor, we have found your slides here, so we are able to reproduce locally. You have just to let me know when wants to change the slide.
Yes. Perfect. It will go fast, I'm telling you.
Can you confirm you're able to see the slides?
Thank you so much.
Take your time.
Yes, I am.
Perfect.
So thank you so much. I'm sorry for that. And thank you for this opportunity to share. So really, I want to provide an overview. There's so many different ways to look at organ regeneration, as you've seen already. Next. So I will talk about the work we do at the LOEX. The LOEX is an academic center, which is important to say. It stands for Laboratoire d'Organogenèse Expérimentale. And we've been operating since 1985. And we're located in Quebec City, 3 hours away from Montreal. Next. And really, we are the biggest under one roof tissue engineering entity. And we do look at regenerative medicine from the tissue engineering perspective, you know, mainly when you have a bigger defect. Of course, we want to use the tools we develop as in vitro tools, but as you will see, we are already in the clinic for some tissues that we reconstruct that have been grafted to patients. Next. So some of the tissues are indeed aimed at replacement, but some of the tissues that we reconstruct can also help stimulate endogenous repairs, such as dressing for skin. So here you have an overview of each of the member of the LOEX. We are around 10 PIs now with our own team. And we, as you see, we are interested in many, many organs. We are specialists of the urologic tissues. My team is working on fat and soft tissue and bone, as you will see. But like I pointed out, two of the tissues that already are grafted to the patients are the cornea for the eye and the skin for the burn patients, especially. And now I lost my slides. They are very tiny, but I guess I can go on. Next. The core technology that we share among all these tissues are what we call the self-assembly of tissue engineering. So it's a very natural way. The extracellular matrix is not provided exogenously like with chitosan from shrimp or else. It's really we stimulate the cells to organize and secrete their own extracellular matrix. Following long-term stimulation with ascorbic acid. Here you have the first application in 1998 already of a completely biological tissue engineered blood vessels. But as I've shown, we can reproduce a lot of tissues. And my colleagues and I are working either with primary cells or iPS-derived cells. Next. So this very easy way and natural way of producing tissues, you can see those tissues over the last 30 years have been produced at the LOX, from skin to ligaments to some models of early cardiac valves and bladder. And like I mentioned, the beauty is that we stimulate endogenous processes. So you can even think about it a little bit as we produce big organoids. Next. So these models have maybe the disadvantage of not being high throughput a lot, but they can serve as model. And here, since I've known that some of you are interested in extracellular vesicles, I cited Dr. Véronique Moulin at the LOX, who is also working on that. Next. So among my work from my team, mainly we focused on adipose tissue as a source of cells for regenerative medicine, doing adipose tissue engineering, skin, bone-like tissues, and biological dressings, which are not grafted, but mainly deposited in a temporary fashion on wounds, for example. We'll keep the discussion for later, but I'll be happy to talk about how we can add microvascularization to human reconstructed adipose and bone and skin tissues. And we can also, I won't talk about it much, but just to let you know that we have used in the past those reconstructed tissues as tools, for example, to evaluate the impact of inflammation, TNF-alpha, IL-1 beta on angiogenesis. And for those interested in urology tissue or heart valves, I do have collaborators working on that with me. Next. So if I focus just a little bit about adipose tissue, very simply, we use the subcutaneous fat as a cell source for regenerative medicine, and I will call them ASCs, adipose-derived stromal stem cells. They are multipotent cells. Next. But what we've learned in the recent years, as many of you know, next please, is that these cells are not only good for as a source of mesenchymal cells, but they are also associated with multi-potential differentiation, which is really good for tissue engineering when you want to rebuild different type of organs. Next. So in addition to their multipotentiality, also the fact that they do produce a lot of extracellular matrix, which is good for our self-assembly method. As you know, Dr. Kaplan has described it a lot in the last years. These cells also secrete a lot of molecules, whether it's growth factors, pro-angiogenic factors, and of course, EVs. So that's why we want to use them, and they are so important to my program. Next. We can see different type of arrays of molecules that are secreted by these MSCs that we do believe these cells, if we include them in the tissue engineering tissues, will act as a little factory continuously producing these factors. Next. So one of the ways to harness this potential for secretion has been for us to produce what we call biological dressings from ASCs embedded in their own matrix to secrete a lot of molecules endogenously to favor diabetic wounds and also radiation chronic wounds, which are very important skin inflammation diseases. Next. So here, just an example on how the first work of my team looked at adipose tissue. Connective tissue is interesting too, but adipose tissue, surprisingly, there is a need for it in reconstructive medicine. And you can see here in the middle row, in the bottom row, that we can reproduce. We can, at the same time, make the ASCs produce matrix and have them differentiate into adipocytes, which are round and filled with lipids. Next. So these models, we can then go even further, making them a little bit bigger. And since some of you are interested in imaging, next, we've been able to graft those tissues and follow non-invasively their behavior using MRI with a collaborator. So I think this is the future, to be combining non-invasive imaging with tissue engineering as well. Next. I won't say too much about the vascular components. We'll discuss it later, next. But just to show you a few examples of how we can precede our tissues during engineering with endothelial cells, and these cells self-assemble to become blood vessels. Next. As you can see here in green on the left and in red in the middle, those are reconstructed capillaries in reconstructed adipose tissues. Next. This is, we can skip this one, thank you. When we graft them, they interconnect. While the slide is changing, I have time to describe it a little bit. We detected murine red blood cells into those human vessels. Next. It didn't change yet. Yeah, so in conclusion, not too bad. I respected my time. We can reproduce so many tissues. I've spoken mainly of normal human models, but we have some colleagues working on psoriasis. They can reproduce the skin, the diseased skin in vitro, to then look for molecules to help it. Of course, we are interested in stem cells because tissues like skin, they renew each month. We need to be able to assess them. We've shown that we can keep those cells in our models. And of course, we can include the degrees of complexity. I've shown you the basis, but we can add more cell types, more structure, and I'll keep the rest for the discussions. If you are interested, there are positions available in my team. I want to take this opportunity. Next slide. To thank all the people that have done it in the fun. And finally, next slide, just a few mention. If you want to note the website of TECEL, for a few months, I am still the director of the TECEL network of the FRQS, the government of Quebec. Next. Which is the network for cell therapy, tissue engineering, and gene therapy. So please go to our website. Next slide. We do have and support 2 platforms of IPS and gene editing. So if you want more information, please write to me, go see the websites, and we'll be happy to help collaborations between Italy and Quebec. Thank you.
Thank you, Dr. for that. Thank you very much. Well, we can come later on after 10 minutes break. See you soon. Welcome back, everybody. Hope you have enjoyed the break. You will enjoy the next phase of this session, which will open the discussion between our precious speakers. And we can actually start from Professor Piccolo, who also has to leave a little bit in advance. So, Professor Piccolo, we found, of course, very interesting what you have presented in your previous presentation. And let us just express some curiosity more than real questions about your discoveries and publications. Also, I would like to include this kind of consideration within the decellularization processes that also we have as our practice, as research practice in the Life Lab program. Several groups work on extracellular matrix after decellularization. I would like you to tell us, what do you think, though, about the relationship, if you can explain us the relation between the aging, senescence, and regeneration, and if actually the extracellular matrix can represent, as you mentioned before, the culprit for aging. You can use this one.
Yeah. So my view is that obviously there is a connection between aging and senescence that is in turn connected to how cells read their context and undergo senescence as, if you want, a way to, people would say that these are zombie cells. I don't think these are zombie cells. I think that these are not something that happens by chance. It's not a default state, but is a reprogrammed fate of cells. Also this one, getting into senescence, it's something that recapitulates once again, in fact, a problem of tissue regeneration. Because during regeneration, there is a requirement for senescent cells. With no senescent cells, there is no regeneration. And the reason is likely that these cytokines that we have a hard time to understand, that is connected to the senescent secretory phenotype, also called SASP, orchestrate the behavior of a village of cells, including, for instance, immune system, right? So the connection between activation of senescent cells and immunity is very intimate. And these may be connected, for instance, to induction of tolerance. It's known that senescent cells can induce Tregs, that are, the quenching system on innate immunity. They control autoimmunity, inhibiting it. And very likely, this is part of allowing into this microenvironment the development of programs that may be highly immunogenic. Okay, so it's all a continuum of event. And these cells are not, certainly zombie cells. Now, beyond regeneration, in which senescence is key as a part of the package, senescence, of course, once it is continuous and prolonged over time, it's inducing aging. It's a status not of acute, but rather of chronic inflammation, that not by chance is associated to innate immunity suppression, okay? And adaptive immunity suppression and innate immunity activation. Now, what is particularly appealing, I think, is that during aging, where you have such accumulation of senescent cells, you may create a hot microenvironment that can contribute to a systemic inflammaging. Of course, this is not something we discovered, but that may also fuel tumor emergence. That may explain why cancer is such an aging-related and senescence-associated problem. And these also include, once again, immune suppression. So there are several hallmarks between regeneration, aging, and cancer that are connected with each other. That's our view. A very seminal work, of course, using mice in which you can selectively ablate senescent cell as soon as they come up, shown clearly that you have, if you could be able to target these senescent cells as therapeutics, you would, massively increase healthy lifespan. That's what they. Maybe you don't increase lifetime, the extension of life, longevity. That's what they call. But you may be able to extend a healthy living at mid-age, for instance. There are intriguing approaches to do that in vivo. Now they are using these CAR T cells. You may know CAR T cells in the cure of some immunological malignancies. They are kind of gene therapy modified cells that express a specific receptor that attack, disease cells. Well, you can engineer CAR T cells against old cells, against these senescent cells. So you can have an immunological clearance. And this is actually increasing the phenotype, improving, if you want, the phenotype of that is aging-related traits in some organs. So there are ways to intervene with this. There are drugs that are called senolytics. So this is actually a very intriguing area. It will be interesting to know whether these impact on regeneration. My expectation and this will impact on regeneration. Another open issue connected to this is, when is the critical time of aging? So aging may not be a continuous event, a linear event. Aging may go through waves of crisis, and maybe hitting those crises with senolytics, maybe you don't need a lifetime treatment, but you need a short-time treatment. So it's a very exciting area on how to rejuvenate yourself, how without affecting your ability to regenerate.
Here you are, the new one.
Yeah. The concept of inflammaging, I think, is well accepted. But do you think that aging also can be a process outside, not related to inflammation? Because there are, for example, different types of death for parenchymal cells, for example. And so I am asking, do you think that can we foresee something different?
Is there aging without senescence? My guess is yes. It depends how you define aging. So, of course, if you have any condition that would increase cell death, in context that cannot regenerate, is clearly inducing tissue degeneration and death. And yeah, but it's a kind of a subtle question what you ask, and I completely agree and see the point. Of course, the emphasis in recent years has been on senescence and inflammaging, but I agree with you that there maybe, let's put it this way, sublethal conditions that are not senescent, and there can still be, age accelerating. I think that's very true. Maybe immune mediated, right? So the thymus is clearly declining during aging. And this is kind of shifting immunity from cell tolerance to peripheral one, which increases, because we are like a bomb, ticking bomb. Without, controlling the immune system will eat us up alive, literally. And so we need a lot of immune suppression. That's the fact. Maybe every differentiated cells of our body has tons of innate systems that keeps immunity at bay, and tons of Tregs, likely. So any imbalance in this, and this is clearly the thymic degeneration, is obvious and known since ages, I think will impact on aging. That's one thing that is not the obvious senescence stuff. Okay, certainly what we show is that senescence need to be focalized on stromal cells, and that senescence of stromal cells is sufficient. So we don't see much of an effect in epithelia, which is interesting, because stromal cells do not renew. Fibroblasts are very long-living cells, and they are hardly duplicate. And even if you have a damage, if you look at the fibroblast behavior, they never proliferate to compensate the wound. They actually becomes much more stretched. They prefer to stretch out rather than duplicate themselves. Okay, so these are very slow cycling, if anything. So so this is why perhaps they like to undergo senescence. I'm not sure about senescence in epithelial cells. I mean, as far as we can tell, many epithelia can, have proliferative and differentiation potential, able to cover many lifespan. Like, look for instance to the experiments they have done for the skin, right? From a tiny patches of skin, you can grow meters and meters of skin in vitro that is totally functional. And that means that every stem cell, can maybe sufficient for a lifetime. So we have much more stem cells that we need, for instance, right? And this is likely true for perhaps many epithelia. I think the organoid are telling you that under appropriate conditions, self-expansion of epithelial cells in a completely normal way, without mutations, without cancer, without transformation, is absolutely a reality. So we are not limited in epithelia. I think we are limited in what is surrounding the epithelia, which is unfortunately what is missing in all organoid cultures, which is stroma. I think the stroma is the culprit of aging, not epithelia.
Thank you. Any questions from the Canadian speakers? Please, Dr. Christ.
Yes, hello. I hope you can hear me okay. I have a question for Stefano, because you were mentioning that senescent cells are important for the regeneration of a tissue, partly through the cytokines that they produce. And so targeting them therapeutically could be counterintuitive if you're eliminating these cells. So my question, are there good senescent cells that you want to keep and bad senescent cells that you want to get rid of?
Potentially, yes, potentially. That's very true. I think, in principle, this is an argument that maybe a subpopulation of senescent cells, discriminating the good ones from the bad ones. It's a very human way of stratifying things. I think that in vivo, this is way more plastic and way more gray zone than we would like to be. So I'm not sure, and I hope to be, wrong, of course, that this simplified, elegant idea you have, I fear it's not going to be real. But it's true. It's true. I mean, I'm not aware, for instance, of any single cell analysis done in living tissues on senescent cells, and that can discriminate these two populations that you hypothesized. The truth is that most of the senescent studies have not been done, upon injuries that are physiologically relevant. People have been studying DNA damage, radiation, mainly in vitro. Very few studies have been conducted on what is inducing senescence on real tissue in real life. I doubt that this is DNA damage. I doubt, because DNA damage, needs to be substantial to trigger senescence. Maybe, connected to something you don't think about, which is metabolism, bioenergetics, and even, excess of mechanical challenges can induce damage because it basically breaks the nuclear envelope. Defects in mitochondria, bioenergetics can trigger the damage. So, I mean, there has been, unfortunately, very few studies in vivo on physiological causes of senescence, because you would need to study also senescence over, a lifetime. When do you pick the mouse to make this assay? At 7 months, at 15 months, or when? And the cell at any given time, the number of senescent cells are so few. So there are technical limitations to make that. So, but I think it would be fantastic to be able to know what cytokines can be a marker for the two cells.
Any other question?
About the relation of neoangiogenesis, vascularization, and the senescence and aging.
Yeah. Again, this has never been dissected appropriately, and you may think, that changes in oxygen levels, even subtle changes in pH, oxygen, and mild hypoxia, can trigger all sorts of defects, and then prolonged can be an acute damage that leads to senescence. For instance, if you have an excess of oxygen, this may induce damage in DNA and mitochondria, and this can be in turn very easily sensed as a damage with activation of innate immunity. That is the ultimate cause of senescence. So senescence is nothing spectacular rather than the activity of two transcription factors, NF-kappa beta and IRF3, and what is inducing these two transcription factors is, in fact, nucleic acid sensing, rupture of chromosomes, and potentially this type of metabolic, if you want, damage that affects mitochondrial permeability.
The other aspect that I think is challenging is always the idea that aging in the living body is different in different organs.
Absolutely.
So this is also something that is puzzling, because if we are thinking to a sort of a clock that govern our aging, then we should expect to have a similar process. How can you couple this with the idea of, for example?
And I can add to that, however, that even if you have one organ that for some intrinsic reason can age very fast, very rapidly the rest of the body will age. There is a way to harmonize aging in the rest of the body. So it's true that you may have a very old muscle on very old skin, but rapidly you will age in the remaining part of the body. So there was a beautiful experiment done, for instance, in mice, where they knocked out a gene that is important for the skin, for the epidermis, this P63. And not only the skin aged, and it was knocked out only there, okay? The rest of the body was completely healthy and fine, wild type. But very rapidly, even bones and muscle catch up with aging. So as if there are systemic mechanisms, likely through hormones or inflammaging, in fact, in which senescence in one tissue induce senescence in other cells, as in a domino effect, you will be, aging very quickly sometimes. So this is exciting to me, yeah.
If I can add, and then I will stop, there is also this challenging aspect that the brain sometimes seems not to go into aging all the rest of the body, as you say, skin, muscle, and so on, parenchymal organs, but the brain seems to, in some example,
remain younger.
No.
Let's hope. Or vice versa.
Maybe we started where the brain.
Okay.
We cannot really have questions from the audience, but a couple of them, if you do have them, we can make it. Any question from the audience? It seems nope. I just want to express a curiosity about the methylation of the DNA as a marker for aging. What do you think about that? I mean, I'm telling you this as we, as a group, recently had a chance of publishing a paper on Scientific Report where we analyzed the methylation of the DNA as an index of the aging. And actually we found, definitely is a preliminary data, so we can still have to work on, that a myocardium of healthy myocardium derived from otherwise healthy donor in transplantation, so human study. Actually, we had a sort of difference between epigenetic and chronological age of almost 14 years. Telling you is a kind of preliminary data, but we had reproducible ones in 15 patients. I was just curious to understand if you can consider the DNA methylation a real marker to measure aging.
Well, yeah, there are this way to count in addition to telomerase length. And
yeah.
There are many ways of counting aging and recapitulating aging. Possible. It's also connected to epigenetic in terms of chromatin structure. Likely, all this reflects maybe changes in nuclear chromatin organization. There are areas of heterochromatin that are normally very packed at the nuclear border, and these areas are clearly affected during aging. So changes in the genome packaging may have a secondary event, changes in DNA methylation itself.
Okay.
But I'm not an expert.
So it can
more
be
than that.
an epiphenomenon, you mean?
Well, it's a marker.
Yeah, it's a
so
marker.
As good as the others.
Thank you, Professor Piccolo. Thank you.
Pleasure.
I would like to move on in the discussion, going a little bit back on what our Canadian experts has presented to us. And particularly, I would like to focus on the endogenous healing through the satellite cells. I had the chance to see that also Professor Angelini has talked about satellite cells in the myocardium. And one of my curiosity, I'm telling you a question would be too much, but is, what is the, your idea, where did it start? The idea of using endogenous stem cells instead of reprogramming or differentiated other kind of IPS, as for example in the skeletal muscle. And if you can envision, Dr. Chris, in particular you, but also all of you can definitely help me in this. How do you envision the future of this strategy? I mean, have you ever thought about other potential clinical application or in other muscles, for example, the heart yourself?
I can begin if you like. I have a slide that I could share to show endogenous repair in skeletal muscles. So I'm going to try to share my screen again.
You mean your presentation, right?
Yeah, it's in my presentation, but I'll see if I can share it again. I think you might be able to see my screen now on the right-hand side.
We can, definitely we can.
Okay, great. So the idea about promoting endogenous repair came about by multiple studies now that indicate that the progression of muscle disease is associated with a decline in both the numbers and the function of the tissue-resident stem cells, like the muscle stem cells. And so on the left-hand side of this figure, I'm showing you what occurs during robust repair of skeletal muscle. That after an acute injury, the muscle stem cells become activated and rapidly proliferate in order to expand the population of myogenic progenitor cells that are then needed to differentiate muscle. And achieving the balance here is critical for the regeneration of muscle, because if you have precocious differentiation at the expense of expansion of the myogenic progenitor cells, then the fewer cells that are available to regenerate the tissue will result in less efficient repair. On the other hand, if you completely block differentiation, then of course, intuitively, you will not facilitate repair. Now, these days, in the context of both a disease like Duchenne muscular dystrophy and also aging, so the muscle wasting that is associated with aging is called sarcopenia, it's been shown now that those diseases are tightly associated with a decline in both the numbers and function of the skeletal muscle stem cells. And so here on the right-hand side is a graphical abstract of what might occur in a disease like Duchenne muscular dystrophy. And it's been shown that muscle stem cells in the context of this chronic degenerative disease begin to lose their proliferative potential. And so that results in a lower overall population that's required to efficiently degenerate the muscle. And up until now, a lot of the strategies to treat a disease like Duchenne muscular dystrophy or in aging has solely focused on the skeletal muscle myofiber and strategies to increase its strength and its mass. But this idea that the myogenic progenitor cell population is depleted in muscle disease represents an important vulnerability that we think we can target therapeutically. And so I'm going to stop sharing my screen again. And just on that point, trying to identify the molecular mechanisms that regulate muscle stem cell expansion potentially reveal new therapeutic strategies to be able to increase the number of progenitor cells in vivo that would then participate in more robust regeneration of muscle. And I would expect that that type of strategy might delay the progression of a disease like Duchenne muscular dystrophy, but also be quite relevant to aging muscle, where we see a loss of the muscle stem cell numbers that in sarcopenic muscle.
Coming to the heart, our experience is that we have niches, especially located in the epicardium, where there is this transdifferentiation, mesenchymal telial cell transdifferentiation, while the niches in the heart are usually not found in the endocardium. So I think this could be related to some mechanical aspect that we have not considered up to now. But this is, we had some data for example, in some models of remodeling of the ventricles after a surgical procedure. And we have seen that when we damage the epicardium, the amount of fibrosis will increase and we will lose the parenchyma, the cardiomyocytes.
And yeah, perhaps connected to this, about the general topic on how to grow, progenitor muscle cells. You have been using about Professor Colin, Chris, this regulator of translation. But similar events have been described, I don't know, I didn't follow up much more, by Helene Blau, I think, several years ago in Science, using specific matrices, biomaterials, using specific with defined stiffness. So once again, she used biomaterials that are different from tissue culture plastics and approximating the stiffness of the real muscles. And she was able to grow similarly to what you are doing now with an inhibitor on plastic. So I'm wondering whether translation and mechanic signals can be in fact two sides of the same coin. I don't know whether you had any thought about it.
I'm actually interested in this topic because what we see in quiescent muscle stem cells is that there's a global repression of protein synthesis. And so then the question is, how does the cell sense that it's within its niche to maintain low levels of protein synthesis? And we've started a new arm of research in our lab looking at the activity of adhesion G protein coupled receptors and how signaling from these receptors potentially impact translation through, or protein synthesis through these pathways that globally repress protein synthesis. And then with respect to the ex vivo expansion of muscle stem cells, so you're right that Helen Blau's lab showed that this could be very nicely done on matrigel surfaces, not matrigel surfaces, but on soft substrates where needed the correct surface tension. And so it would be interesting also to see if the combined aspect of our small molecules with these soft matrices could further expand muscle stem cells ex vivo.
Thank you, Dr. Chris. So what about the imaging modality and advanced imaging modalities for stem cells, quiescence, or other kind of IPS, Dr. Darkan? I've read you have developed advanced imaging modalities for those.
Yeah, so can you hear me okay?
Yeah, we can hear you.
Okay, perfect.
Perfectly.
Yeah, so it's kind of, we've gotten into different types of imaging because in a way, when we started with organoids, it was always, how do you image through tissue of different thickness? So it was always originally just slicing through it at a very fine amount. And I guess more kind of over the last few years, we've gotten more into like tissue clearing for at least fixed tissue. And then I guess the last year, actually coming up into this year, we're actually starting to explore more like how do you do more stuff with the live? Actually, it's led to a more collaboration with Laval and kind of Julie's area, like with Marie-Ève Paquette and Yves de Conic, more looking at AAVs and biosensors, because fixed organoids going to give you a sense of one point in time. Biosensors, we're kind of exploring, could we do this stuff more longitudinal in living tissue? And it's not just applicable to brain, like we're actually we're exploring this. Could it be applied to muscle? Could it be explored to other tissues? And I know there's others kind of looking at kind of 2D optogenetics, the dreads, different G-camps that you can then look at activity-based stuff over kind of a longer term. But I think the imaging modalities will be kind of the way, I guess, that we can go from cardiac cells to brain to muscle and kind of really kind of tease out a lot of the stuff and get a lot more information when we combine it. with some of the other slides, maybe on the multi-omics as well. One of the areas we're quite interested in as well is, the waste we throw out. We never talked about it today. Like the organoids we make, we throw out a lot of media, and it has a lot of the goodies and factors that we always wanted to look at. So the nano factors and EVs, we've actually done some work where we've kind of pulled EVs out of organoids, and we can then show the stuff in there. And then when you kind of look at the organoids and then you start looking at the secretomics, then you really start to get this kind of picture of patients that you wouldn't have even had before. So I think it's a very fruitful area, very unexplored, a lot of gold there to be found.
Maybe I can ask.
Yes, please.
To Thomas something, since we are, very intimate here, and let me ask you something very direct. Because I've been always kind of surprised about these brain organoids, to be totally blunt with you. Because these are, I've seen some, I've never used them myself, but how real are these things because, I've seen some histologies. I don't know whether these were imperfect or something, but seems to me like a mess. So there are clearly segments of organized tissues. I'm not sure whether these are fetal things or at 12 stages of development. But, patterning is,
No, it's a great question. It's always
I
a
have
great
no
question.
idea. So it's a lot of mix-up of things. And although these are, something that you have to use in a sense at the same time, you need to be very cautious, or I think we need to be very cautious, that conclusions out of these extremely heterogeneous, complicated, God only knows type of things can be, also very questionable.
I think
Before
you
you
kind of
answer,
hit the...
Dr. Darkin, I just wanted at the end of your intervention, Dr. Fradette, to add some cues with the vasculature, as we mentioned before. How the complexity of organoids has changed after the inclusion in them of the vasculature system.
So, yeah, so
Please.
So I feel that the field was very much driven by this beautiful picture that, Knöblich had of this, that looked like a brain in the Lancaster Nature paper. But that is truly a very good section. Whether that's real, it's, and I started questioning myself after I see all the time the same picture. I say, you see what I mean?
I know, I really understand. I think when we started on this, we were kind of skeptical. Could you make a piece of tissue similar to what you might have at the midbrain? Because we were interested really on Parkinson's originally, and then people would kind of come back and say, are you crazy? You're kind of growing a piece of tissue for a few months and it's almost early in the developmental phase. You may never see it. But the more we kind of worked on it, the more we started to get the chance to see what the benefits are and the disadvantages are. You've hit the nail on the head with a few things. Organoids for many years kind of came with this caveat of, they're the greatest model ever. They are a good model and we like to use it, but they have limitations. One, they don't have vascularization. Two, they don't have structure, for instance. There's no up, down, left, or right. And they are actually, if you ever put your finger down on it, they're a lot more squishy than if you ever touch, let's say, a human brain or a mouse brain. And that's kind of an area that needs to be kind of worked on and really developed. How do you actually introduce more structure? How do you introduce it so it's a lot more physiological? I know there's been a lot of hype from Motter's group where they show the whole, you grow an organoid for 8 months and you see brainwave activity, and then that led to articles, saying could a brain organoid be somehow have a feeling? I don't personally believe that. I think, it's going to be a lot more, iteration over the next few years to get to that. But I don't think we can discount them as a useful model. I think that's when we kind of have a midbrain organoid and we actually do single cell and you start to compare it to what you would have in the substantia nigra, a lot of the cells you would have are actually quite similar in terms of number and composition. I think they are missing microglia, which is a limitation as well, but I think that's kind of where we're at. So I think they have a lot of advantages because you can grow them much, much longer than flat 2D cultures. They tend to have a lot more of the heterogeneity that you would expect of a brain tissue, and it's human in nature. You can get a lot more of it. So I think you can get a lot of insight into the disease, and as the methods kind of develop and the way we characterize it develop, I think we can start to get more and more into that way where, you know, it's a lot more advanced. And I think the one maybe crux is organoids came out in 2014, like 2013, 2014, Madeline Lancaster and Jürgen Noblik, they had that initial paper. So the field is not even 10 years old. Whereas when you look at mice, they've been going for a whole century. They have a very impressive CV. So I think we have to give the field a bit more time maybe to mature, to get, you know, in 2 decades, we could be a lot more advanced. But we have to kind of obviously have that kind of asterisk to kind of, you know, take note of what the data might be.
Second question, well, before the second question, let me keep the record straight. The first organoids in brain were not done by Knoblich, but by
that's true.
My mentor, Yoshiki Sasai. That needs always to be remembered. Okay. The other thing is about this idea that is you read all the time that you can take you know, patient-derived from disease and get organoids and get human tools to study drugs. I think this makes a lot of sense for genetic diseases. But I have really hard time to appreciate how can this be useful, taking a patient from a fibroblast, from an Alzheimer patient, and then getting brain organoid from this patient. If you can start from a fibroblast, I mean, the brain organoid that you are going to have is going to be a brain organoid, irrespective of coming from an Alzheimer patient or a Parkinson patient, unless there are genetic roots of these diseases. So because the reprogramming through epigenetics will be, you know, like a complete washout or whatever epigenetic imprints you have that are non-genetic. What do you think?
No, it's an interesting question. I think the familial diseases, like, you know, with Parkinson's and Alzheimer's, a lot of the genes help kind of point us to kind of conserve pathways, for instance. And that kind of lets us give it an introduction into that. I think in terms of, you know, can we go from fibroblasts into neurons? And it gives us an idea of what's going on with the disease, for sure. There's been some great papers that have come out on that. And even on the PD side, like Janelle in UDEM, and there's several other groups as well, kind of doing direct reprogramming from fibroblasts to neurons. But in a way, they discount all the other cells. They're focused on dopaminergic neurons or hippocampal neurons, and you forget all the other cell types that are there. I think that's one of the areas you can really capture with organoids. You can start to bring in a lot of the glial subtypes. You can co-culture them, like you can add them and mix them together. But there's been quite a few studies coming out from, I think there's been groups around in Europe where they've started to look at iPSCs into neurons and other cell types, and they're starting to see kind of conserved features in bioenergetics, lysosomes, synuclein pathways, and a number of other pathways that kind of help us actually point to where we can actually maybe get with sporadic patients. And I don't know, we've talked to some of the clinicians and sporadic PD, for instance, like maybe it's just we just don't know the genetics yet. Maybe there's other kind of features that maybe we don't know yet. But I think for now, it's kind of we're stuck with what we have in terms of familial kind of patients. Cell lines will help kind of point us to potentially conserve pathways. And then we can start to examine with like sporadic or idiopathic patients, is there an additional conservation, or maybe it's completely not. But from some of the data so far, it seems that lysosomal dysfunction that has come from the GBA gene with TD, lysosomal dysfunction seems to be kind of conserved across a lot of the patients with PD. And similar with immunoinflammation, like there's issues with PINK1, there might be a whole conserved pathway there in inflammation as well. So I think we have to be, it's like everything. Organoids have limitations. Studying familial patients has their advantages. It has its limitations. But I think, you know, if we keep studying it in the PINK1 knockout mice for 10, 15 more years, we're probably just going to be where we are now. So we have to try and do new things.
Absolutely. Thank you.
No problem.
Dr. Fradette? Do you have any comments
Yes.
about that, particularly about the, yeah, please.
Yes, well, I think you specifically asked me about adding more cell types, like such as endothelial cells into the structures. So, of course, size is very important. So, I think adding vasculature, like to organoids, will really help to understand the communication between the cells. We can also like recreate a kind of blood-brain barrier, and also, but of course, when it is most needed, it is when the tissue is more voluminous. So that's why maybe I will talk just a little bit more about our self-assembly model that can give rise to sheets. So in our models, we just include those endogenous endothelial cells during engineering between our sheets that we can then pile up and make bigger tissues. And what we've shown and what we've seen is that very naturally, these endothelial cells do recreate little capillaries by themselves. This has been proven to help inosculation after grafting. For example, if we build a skin or fat that contains this prevascularization in the tissue, we can see that the vascularization of the reconstructed tissue will connect with the host vascularization is as soon as 4 days, which will be much longer, up to 10 days, 12 days, if there's no prevascularization. And we know that the survival of those cells, it's critical to have a nutriment, early vascularization. So I think we've shown that adding such a component, this vascularization, is really necessary and very good using those in vivo grafting models. I think the next challenge now, and if people want to collaborate, and I've heard the word microfluidic in Thomas' presentation, I think the challenge will be to use and pursue those in vitro models that contains the vascularization. For the moment, there's very few models that helps to have a microfluidic, to have those flux into the reconstructed tissues. And I think if we go further that way, it will really help to understand all the metabolism and how to keep the tissues in vitro and learn more how the cells communicate and what benefits it can bring. But definitely when you're talking about bigger tissue, or maybe like it was mentioned too, like an assembly of distinct organoids, we'll need to go with flux circulation conduits. And if those conduits are lined with endothelial cells, then we can talk about prevascularization.
Any other question? Any other comments from the audience? Yes, Professor Gerosa.
Let's go back to senescence. I have a very naive question for Stefano Piccolo. Let's say a very surgeon question. So if within a system, a part of the system is becoming senescence, if I understood well, all the system will become senescent. Is that correct? Somehow? Yeah. So if I'm transplanting a organ from a younger donor to an older recipient, I would expect this organ to become senescent.
Yeah, of course.
Yeah.
It's hugely complicated.
Yeah, sure. I said it's a surgeon question. But now, on the other side of the coin, if I transplant an organ from an older donor to a younger recipient, what's going to happen? The entire body will become younger, or the organ will become younger? So
I think that the second, unfortunately, the second one is going to be more true.
So that's mean if you're implanting a older organ, an organ from an older donor to a younger recipient?
I think you will. I mean, this is something that we can only speculate.
Yeah, sure. It's a very provocative question.
And if let's remove also from the equation the damage of, of the immune system.
Ischemia, reperfusion.
Rejection,
Yeah.
which will be a super
Yeah.
aging impact, a huge aging impact. So let's imagine a pure theoretical way that you have a transplantation from syngeneic two siblings, from one to another, at different ages. Let's put it
no
this way.
ischemia, reperfusion, all this stuff.
Whatever, okay. It would be interesting. Who wins? Let's put it this way. It would be interesting. I mean, there are clues that, as you know, that through this fusion of the blood system, you can rejuvenate an old body with the blood of a young. Okay, it's called parabiosis, which is not even legal to do this experiment in Italy for connecting the vasculature. So, so that's one thing, and clearly in vitro, old niche induce young cells to become old, sorry, and vice versa. A young niche can rejuvenate old cells. I mean, there are experiments. So I think depends on a balance. Let's put it this way. Extracellular vesicles are also interesting in this respect. Are they rejuvenating or aging extracellular vesicles? We don't know. I think these are complicated experiments to do. I think you need to do it first in mice, where you can control the fact that they need to be syngeneic and provide proof of principle there. And yeah, that's a nice thought.
Okay, thank you.
So we are running out of time. It's supposed to end up today at this time. So I want to thank all our speakers, the Italian present here and the virtually present from Canadian. Hope you have enjoyed the discussion and the meeting. I'm inviting you for the meeting of tomorrow focused on genetic disease in rare disease. And I hope you will be numerous as for today. Thank you very much. See you soon.
It was a pleasure. Thank you.
Thank you for coming.